All right, so hi everyone. Good morning or good evening, I guess, depending on where you are. So we're really excited today because we have a guest speaker, and this is Dr. Aivett Bilbao, and she is coming to us virtually from PNNL, the Pacific Northwest National Laboratory. So Dr. Bilbao is a computational scientist with the Systems Modeling and Computational Science team in PNNL's Environmental Molecular Sciences Division and the Environmental Molecular Sciences Laboratory. Now she received her PhD in computational mass spec from University of Geneva, and then she went on to do a postdoc at PNNL, where she currently is now, developing a number of computational tools for mass spec-based omics. I think working on both proteomics and metabolomics. She specializes in data-independent acquisition and tools for that. And so we're really excited to be learning about that today, since I think many of us in the audience, at least myself and Daniel, mostly do DDA, data-dependent acquisition. And so, yeah, super excited to hear what Dr. Bilbao has to say. And so then without further ado, I will turn the floor over to her now. So I hope everyone enjoys, and we'll be taking questions at the end. So please save your questions for the end, or you can write them down in the chat so you don't forget. But yeah, without further ado, Dr. Bilbao.
Thank you, Allegra. Thank you, and Daniel, for this invitation. I'm excited also to be here and have a good exchange of ideas and discussions with everyone. So nice to meet you all. So today I would like to start talking about data-independent acquisition, mass spectrometry, some basic concepts and difference compared to the classical data-dependent acquisition. Then I will go over some of the software tools that are available for metabolomics. Next, I will go through talking about ion mobility, because that's another of my areas of expertise, and how we combine ion mobility also with DIA into multidimensional workflows. And then I will talk about Peak Decoder, which is a workflow, an algorithm that I developed to process this kind of data, and then discuss the progress that we have towards developing an improved version. So as you all are familiar with it, the omics mass spectrometry, one of the most common workflows is to use liquid chromatography in front. And the typical data-dependent acquisition workflow, the instrument, it's collecting survey scans, so MS1 scans during each cycle, and then it selects the top N fragments to serially fragment them, and then it collects MSMS in this fashion. So it's isolated one by one of the top N ions, depending on the acquisition settings. In this way, yeah, across the chromatography separation, then we have the MS1 data and we have the MSMS. And the key difference between DDA and DIA is that in DIA, there is no selection of single precursors. We basically co-fragment and co-isolate and co-fragment multiple ions. And the difference is that we can then, from the raw data, build chromatograms also for the fragments, which is something that we cannot do with DDA. There are several variations of for DIA acquisition. One that is called all ions or all ions fragmentation, or MSE, is basically operate the instrument alternating between low and high collision energy to collect the MS1 and MS spectra, the fragmentation spectra. And basically the instrument is not selecting single ions, but we open all the quadrupole of the isolation window to let all the ions pass through, and then collect fragmentation spectra, multiplex it for all those ions. And the other variations, the most recent one, which is the swath, which is basically instead of fragmenting all the mass range of interest for the precursors, we can create a series of windows, which is not as small as in the case of DDA, but we basically open it a little bit to cover the mass range that we want to study. But then the instrument is not selecting a specific ion, it's fragmenting everything that goes through that isolation window. And then the instrument cycles through the same scheme of isolation. So those are the main two schemes. And in terms of instrumentation, there are time of flight, quadruple time of flight instruments to do this kind of analysis, or also in the Orbitrap platforms that we can do also. If we have the quadruple, we can do this kind of SWAT. So the difference is then it's an Orbitrap mass analyzer. And this animation is basically showing the same. The gray symbols, little boxes, are for reference to compare. As I said, we increase the isolation window to systematically fragment all the ions that go through that isolation window. And in this way, we collect fragmentation spectra for all precursors. It's more reproducible compared to DDA because there is no pseudo-stochastic selection of ions. All the information is collected within a single run. And then this also has some disadvantages of consequences, right? So then the drawback is that the direct link between the precursor and the fragment ions is lost because we are fragmenting multiple ions at the same time. So there is no single precursor ion selection. And the data is more complex to process, so requires specialized algorithms and software to process. Also, another drawback is that because we are doing co-isolation and co-fragmentation, the likelihood of interference increase. And then based on these chromatographic profiles, that's the basis to do the annotation in the data for DIA. I will talk more about that later. This is a publication that I would like to share, which is, it shows the advantages of DIA in the terms of metabolomics. So we can see here that using DIA, we can detect more number of features, collect more MSMS, and annotate more features in considering including also MS2 or MSMS matches. And the quantitation is also better compared to only DDA. Here, we would like just to show like how also DIA was specifically demonstrated. It has been used. There are established software tools and workflows for proteomics, but it's starting to be more relevant also for metabolomics. And as a example of that, it's that now DIA has a separate session only for lipidomics and metabolomics. Also, we can see that the number of publications is increasing per year. I think this one I made it last May, so right now we should have a little bit more than that. Of course, we have more for proteomics because, as I said, it started for proteomics, but now it's really increasing. It's getting more popular also for metabolomics. There are two main ways or strategies to process this kind of data. One on the left side, which is based on targeted feature detection and MSMS deconvolution. The widely used tool is MS-dial. So basically, the tool detects all the MS1 peaks, and then it applies an MSMS deconvolution. It just extracts the fragments that have the same, in this case, chromatography or peak shape profile, the fragment against the precursor, and then this generates a pseudo DDA spectra because it assigns then that precursor to those fragments, and this can be matched against the library. On the right side, we have the other strategy, which is called targeted data extraction, which basically requires a library to extract signals from the raw data and build these chromatograms and then do a scoring. There are some advantages and disadvantages for each of those. In the case of MS-DIAL, as I mentioned, yes, it does the MS-MS deconvolution, and you can import your library and do an MS-MS matching against the library. One of the drawbacks is that in case of complex sample, it really starts to get complicated to do the convolution of those fragments because there are many things that are co-eluting, and the quality of the pseudo MS-MS then is low, and then the matching against the library becomes low. It has a graphical user interface, but it's not that easy to use then for large scale analysis, and there is no fault discovery rate analysis available in MS-DIAL. And here at the bottom on the right, just showing some results, how it increased in this case the number of lipid identifications comparing SWAT, which is DIA, compared to DDA for lipids. Skyline, it employs the targeted data extraction. It's very user-friendly. It has better quantitation results compared to MS-DIAL because it's a targeted data extraction approach. So it could be more selective because you need to specify what are the fragments and the precursors that you are looking for to build these chromatograms. But because it's basically guided, then it can be more sensitive to lower abundant ions. And it has a graphical user interface that is user-friendly, very easy to use. A drawback is that there is no FDR analysis available for metabolomics, and it can be more complicated to use for large-scale processing because the peak detection sometimes doesn't work well, so it can be difficult to do manual curation for all these peaks to say like where the boundaries, where it starts and where it ends. Another tool is the DIA metallizer. It encompasses both the library creation step from DDA data, and then after building this library, you can do targeted data extraction on the DIA data. And this includes also an FDR, a method for false discovery rate analysis. It's a more complicated to set up workflow, but it's suitable more for large scale analysis. The drawback is that it does not support, like in my case, it does not support the ion mobility dimension. So ion mobility is a separation technique, a gas phase separation technique that is being increasingly cooped more and used in omics analysis to separate different kind of molecules in proteomics, metabolites, lipids. And the advantage is that with ion mobility, because it's a property that is related to the size and the shape of the ions, we can distinguish isomers or different molecular classes. And we can determine, or we can compute the collision cross-section, which is then we can use this property also to increase the confidence for annotation, for molecular annotation. And in this example, we can see, yes, as I mentioned, the collision cross-section is a property related to the size and the shape of the molecule. So if different molecules have exactly the same molecular mass, but the structure is different, then the ion mobility will be able to separate. We can see here, for example, we can see a separation for like biological molecules compared to exposomics or halogen molecules. In the case of multidimensional analysis, how we combine then ion mobility with DIA, it's using the all ions or fragmenting the whole mass range, and the instrument is operated in this low and high collision energy mode. But then we have the ion mobility separation between the LC and the mass spectrometer, And instead of having a single mass spectrum, MS1 and MS2 per each LC time point, then we will have like what we call a ion mobility frame or a one ion mobility separation. And then the instrument is operated to collect both, one with the without collision energy, and then the next frame activating the collision energy. And because the collision energy is activated after the ion mobility separation stage in the instrument, then we can correlate the precursors and the fragments based on the retention time plus ion mobility time. This data is very rich, but it's very difficult to process. The traditional tools don't work. So that's why we developed this workflow. Basically, we are combining existing tools with also some scripts to apply machine learning analysis and put together into a single workflow. And we use existing tools. In the first stage, which is to train the machine learning algorithm, we use MS-dial. MS-dial is used to perform untargeted feature detection from the raw data. It means independent of any molecular identity. We extract the features and we take the high quality features that are deconvoluted. So it means we have MS1 features and the associated precursor fragments that have the same profile in terms of time. And in this case, it's the same concept applies if you have ion mobility or not. The difference is that if it's LC only, it will do the deconvolution only based on the chromatography profile. And if you have ion mobility, then it will take into account also the other dimension. So then we export these high quality features. And then I implemented a script to generate decoys. And then from those lists of features that I will explain later how we generate the decoys. And then we take, because from the MS-dial result, we have this group of high quality P groups that we know the precursor mass to charge, the retention time, the CCS, and the fragments mass to charge. Then we can use Skyline to then extract the signals from those, both the targets and the decoys, again, from the raw data, but now in a targeted way. We are using these values as of the properties as seeds to extract the signals from the raw data now in a targeted way. And then we have some peak metrics. These peak metrics then as a vector, in a form of vector, I used as an input to train a machine learning model. In this case, I use a support vector machine. Now, because we have targets and decoys described by this set of XIC extracted ion chromatogram metrics, then we have basically true and false peaks that the model can use to learn how a good coelution, the scores should look like for a P group that has good quality or high quality compared to something that is just random a set of scores, because those are the decoys. So then the model can learn to differentiate this set of properties. Once we have this model that is trained, we can use then our library. It means you can have a library that could be predicted or built from a set of standards or a public library. And then you have this model that already knows how to distinguish a good quality P group from a bad. So then you can use your library where you have the precursor mass to charge, the retention time, and so on for the fragments of the molecules that you know, and extract those signals to extract all those metrics from all the raw data. And then you can use that model to score. And we can then also do the quantitation based on the peak areas. This is an overview of how the step one, which is the feature finding and MS deconvolution using the MS-dial, just a higher view. You can see that here on the right, we have the blue one. the precursors, and then the multiple fragments that have the same retention time and peak shape. Those are the basics to have a good quality P group. And this is how we generate the decoys. Basically, we have this list of P groups that are high quality. We have a series of criteria. For example, they should have at least 3 fragments. And in this list, all these that are called the targets are sorted in retention time, by retention time. And then we create pairs. We associate these precursors in pairs to create the decoys. And there is a series of criteria also to make sure that this pairing, or to improve the probability that it's at least not a decoy that is likely is too fake or impossible to occur in nature, and is also not very close to the pair that will be created. So they should these pairs like target A and target B. In this case, these are high quality P groups. They should be at least 3 minutes apart. So we are not pairing like the tail of the same molecule. And they should not be the precursor should not be the difference in mass should not be larger than, for example, in this case, arbitrarily said like set 50 mass to charge units. And then once we have these pairs, we swap fragments from the top in most abundant fragments. So then for each of them, we create a decoy. So we have decoy A and decoy B. When we swap the fragments and we re-extract the signals again from the raw data, for the decoys, then we start to see that the peaks and the fragments, they start to get deviated, or the peak width also change compared to the precursor, or there is just simple noise. And then because we use the original abundances also as the expected library intensity, when we do this swapping, the cosine similarity or the spectral similarity, then it will be low for the decoys. This is how the skyline interface looks. As I mentioned, you have to specify the precursors and the fragment ions that you are looking for, and you can extract the signals, and you can export a CSV report with all those peak metrics. To train, as I mentioned, using a support vector machine, and we take these peak metrics that have the targets and decoys metrics to train the SVM model, and I save this model. This is an example from that training data. We can see that each of these metrics individually, they don't do a great job separating the targets and the decoys, but we combine them into the peak decoder score of the that it's out from the machine learning model. We can see that the separation is really improved, and it's better compared to even the cosine similarity, which is the spectral similarity alone. So we built a library with 64 metabolites of interest for this study. After we trained the model, then we extracted the signals from more than 100 samples. And then we could annotate and identify, using this model already trained, we could identify more than 2,600 features. And even for the fragments, for the metabolites that we could not detect any fragments, still using the combination of retention time plus the collision cross-section increase the confidence in the annotation. For the validation, we inspected, visually curated the data, compare like how the standards chromatograms look compared to the microbial sample, the complex sample. We can see the fragmentation profile. It's similar. And Comparing against a manual curation, we saw that MS-DIAL, using our workflow peak decoder, we could identify or annotate many more peak groups, many more molecules. And we also could decrease the number of false positives compared to Skyline alone. We performed dilution experiments for some selected metabolites and compared to selected reaction monitoring. And we saw that using the ion mobility platform, we could reach good linearity and lower level of concentration. This is one of the reasons for this is because in the ion mobility platform, we are quantifying based on the intact precursor plus the fragments. And on the SRM platform, you only quantify based on the fragments that you select. There is no intact precursor. We also ran some of the samples, we ran them by GCMS and the SRM platform. And we saw for the metabolites in common that we could see similar abundance profiles across the different platforms. This workflow was developed in the context of a synthetic biology project. It's the Agile Biofoundry. It's a consortium of national laboratories. The chromatography methods were developed by Natalie Munoz and Yuchen Gao, and Danny optimized the parameter for the ion mobility instrumentation. And in this slide, I would like to show you an example of, we have here the wild type and 3 modified strains of the Pseudomonas putida organism. And we could see in this example, we compare to also the proteomics results. In this case, the circles are the metabolites and the arrows are the proteins, the enzymes. And we could basically see that the modification was producing or activating the pathway that it was designed for. In this figure, it's an example of how combining all these dimensions helps to increase the confidence in the annotation. When this is for each metabolite, if we take different combinations of the dimensions. So for example, if we only take the mass, the accurate mass, and we try to annotate all the features that are detected in those runs, we have many more possibilities, which means it increased the ambiguity. But then when we start to combine the multiple dimensions, it decreases the number of matches. And it's really when we combine the retention time, the accurate match, and the CCS, when we can really decrease and be more confident about this annotation. So this illustrates the power of using the multidimensional separations. All right, so now I would like to show you about the new version that we are working on for peak decoder, and it's not only for metabolomics, but also for proteomics. So one of the main difference between the traditional machine learning methods and the newer deep learning methods is that for machine learning, we need to design what are the features that the algorithm will use to learn. So we have to extract those features. So there is a human there in the loop. For example, we selected, okay, that we will use the mass error, the retention time offset, the mobility offset, the abundance, the fragmentation pattern, and so on. So these are predefined features. But with deep learning, it really has the power to learn from the raw data and has more power to model complexity. And you don't need to specify these features up front. The model will extract those features. So you just need to basically provide the input from the raw data and what is the output that you expect. And the model, of course, you need a lot of more training instances than in this case to make the model be able to properly learn and generalize. But that's the advantage. You don't have to design those features by yourself. And my question and what I'm interested in working on right now is there is a great progress using deep learning and AI in mass spectrometry, but has mostly been for prediction of individual properties. So there is great progress to predict MSMS, to predict the collision cross-section, the retention time, but can we use this model also to learn patterns or detect patterns, extract patterns from the raw data? And we can frame this in the context of object detection in images. In this case, we can take a model, for example, that it already learned how to recognize pedestrians in pictures. We can take this model and apply a technique that is called transfer learning. It means the model already knows how to recognize shapes and objects. And now we will just tune it and tell you, okay, now instead of detecting pedestrians, now we will detect peaks or peak groups. So on the left is the traditional view for the ion mobility type of data, LC ion mobility. So we have the chromatograms and then the heat map for the mass to charge and the arrival time. So now what we are doing is creating a new representation as an image. So we build these two-dimensional plots where we have the ion mobility or the arrival time in the y-axis and then the retention time in the x-axis. And we are building this extracted ion, two-dimensional extracted ion signal or image. So for each of these mass to charge, we can create this image. This is for a selected mass to charge. And then basically what we want with our model is to detect the areas where these signals overlap, because that what it means that these fragments are coeluting and have the same mobility as the precursor, and that's what will indicate the presence of the molecule. So what we are doing is representing these ions as a 3-color channel in an image. So the precursor is red. That we take the top two fragments. The fragment 1 is green. The fragment 2 is blue, and we overlay these images. And basically, when we see these white images, that's what we want our model to detect. These white areas, these white regions, and then the model will detect those images, those regions, and then it will tell us the exact coordinates, and that will be our retention time and arrival time, and also the peak widths. For this, we are using the EMSA data converter. So this is a tool that I implemented to make easier the access to the raw data and working with the raw data. It's a self-contained data conversion tool software that reads the instrument vendor data in that format and generates an output that is a binary output, but it's in the HDF5 format, which is an open format, and you can read in any programming language using generic library. So it means once you convert the data, you don't need any specific special library dedicated for EMSA to read. You can use any library, basically any programming language. And in the current workflow, what we do, as I mentioned, we use the PNNL preprocessor, which is a tool that we developed and maintain at PNNL in collaboration with Agile Technologies to improve the quality of the raw signals. Then we convert the data to the EMSA format. We have curated examples that we use to train the model. We apply the targeted data extraction to generate these image representations of the ions. And then we are working on generating a software executable that will take that model that is already trained, and then we will be able to process any complex data and use it automatically and be able to process any number of samples and use this kind of workflow at scale. Right now, we have applied two iterations so far. First, we trained a model only with 50 examples, and then another one with 200. This is still low, but it's looking very promising. So this is an example using the model V0, the first model. And we have on the left a metabolite example. For this example, the image is, it looks like more square because we do have the collision cross-section in the library, and we could extract, reduce the window where we want to extract the images. On the right side, we have a peptide where in this case, we did not use any collision cross-section, only the retention time. And we are using all the mobility separation. But we can see here that this first iteration of the model training only with 50 images, because these are it's laborious, time consuming to generate this training data. But we can see that the model is able to detect the white regions, the overlaps, even if the probability is low. The model is not very confident. So 0.1 is not really great. And also the model did some crazy stuff, which we don't know what we was training, but it's okay. We only trained with 50 images. But even if the probability is low, we could do a selection of these annotations that the first model did to complete these 200 examples to do then a second iteration of training. And now with this model version 1, we can see that the model is detecting the white regions of interest properly, and is also very confident. Now we can see that the confidence is higher. And this is an example for proteomics. So we are building a data set to evaluate similar dilution experiment also with metabolites, but basically the concept or the validation is the same. We have, in this case, we have 10 a dilution of concentration, different concentration. In this case, this is a reverse calibration curve. We have the endogenous or the light version of the peptides, and we have the heavy peptide, which is the isotopically labeled peptides. And we can see these results are very encouraging because we can see the number of detections that the model could do for the lights, which is the background. It should be more or less constant, and that's what we more or less see. In the most intense concentration point, it's decreasing a little bit, the green bar, and that's it's probably due to ion suppression, because now the heavies are more concentrated. And we can see also that the heavy, we are increasing the number of annotations that it's expected. So we are very excited, and we are working to keep improving it, but this is very encouraging. And this is all fully automated. It's not, we don't need to do a step process to do the convolution first, the detection, and then run a skyline. So this is all fully automated in Python. So to conclude, I hope I could show you that DIA offer advantages for metabolomics too, and it's increasingly used in the field. We combine the retention time, the collision cross-section, and the fragmentation patterns. And from DIA, we can increase the overall confidence in metabolite annotation and analyte identification. This computational workflow and the analytical strategy could be applied to any omics. And because we are learning, we are training the model from raw data, is not depending on specific fragmentation patterns of a specific kind of molecules. We can annotate also and calculate error rates for all omics that we are working also to evaluate and demonstrate. And we expect that Peak Decoder 2, the new version, will be available soon for the community. and will facilitate studies and to provide knowledge in automated fashion, more accurate. So to conclude, I have many persons to thank that have worked in some part of this data that I show you, from all the way from sample preparation and the strains, the modification of those of microorganisms, all the way down to the instrumentation, and then the data analysis, especially to Natalie Mujones and Yuchen Gao, that are working with me closely to validate also the results compared to the other techniques. And Christine has been supporting me to develop Peak Decoder, now Peak Decoder 2. And finally, we are looking for contributions for these special issues. If you are doing any work with ion mobility and AI machine learning, and if you are interested in trying Peak Decoder with your samples, if you have environmental samples, we can talk about potential opportunities because we work I am part of EMCEL, which is a user facility. And also, if there is you have interest in doing a PhD internship at PNNL, please contact me. So now, thank you for your attention, and I'm happy to answer any questions.
All right. Well, yeah, thank you so much for that really cool seminar. So yeah, it's time for questions. So if someone has a question, you can unmute yourself and ask, or you can write in the chat and I will read it out, whatever you prefer. So yeah, are there any questions in the audience?